Hi Tim, how are you?
Hey, I'm doing good. Um, Let me figure out how to add this.
Oh, do you have a
Um,
presentation that you want to add? Then let me.
yeah,
I'm you into a moderator. So now, if you click on these three dots that are under Leave Quietly, I'm not sure.
yeah,
Yeah, there you can change the link by using pinned link, and if you press on that,
okay,
there you should be able to change it.
great. Okay. All right, so that should be available now.
Great, thank you. We'll start in around 10 minutes, so we have time to have a few minutes to relax. I'm sharing it on Twitter and so on that we are starting. Thank you. Hi everyone. Hi Victor, Jocelyn, Arian, Martin. Thank you for coming. Hi Frank, how are you today?
Good evening. I'm fine. Looking forward for the talk.
Right, it's evening already. Yeah, meet Tim, meet Frank. Yeah, thank you for coming.
I'm already sneaking into the presentation here. Just for everybody, if you have an iPhone, and I don't know how to do that on an Android, but you can open the link on your phone and then send that presentation to your computer via Bluetooth. So you have the AirDrop, and then you can AirDrop share it to your computer. And I hope that there's something similar for the Androids. So I'm just looking at that at my computer and completely looking forward.
That's really good advice. Thank you. Yeah, because it's really cool to have the slides big on the computer and then interact through the Clubhouse app on the phone. That's I prefer that too. Thank you, Frank. We'll start in 5 minutes. Thank you everyone for coming. Please raise your hand if you want to ask questions or use the chat to comment or ask questions. You're welcome to do so. That's the cool thing about Clubhouse. You can interact. So yeah, feel free to come up to the stage. Thank you.
I guess another way on Android would be, or any other phone, to open that presentation and then copy the link and then drop that link via some messenger or something to a computer. So we are just gathering, starting in 3 minutes, I guess, Katarina.
Yep, starting in 3 minutes. Thank you for coming and thanks for hanging out with us. I hope everyone has enjoyed their week. Happy Friday, everyone. Hi, Denise. How are you? Happy Friday.
Hi, Katarina. Hi, Tim. Hi, Frank. Happy Friday to all. Looking forward to this talk.
Okay, I think we can slowly start. Yeah, it's 1 more minute, but until we start, it should be time. Welcome everyone to the Science Society, and of course, a special welcome to you, Tim. Can you hear us well? And, yep, there we go.
Yeah, got it.
Just checking. Perfect. Yeah, welcome everyone, and of course, a special welcome to you. Thank you for coming here on Clubhouse to give a talk at Science Society. We really appreciate it. And before we start, let me introduce you a little bit to the audience, and then if it's okay with you, would ask you a couple of interview questions, and then it would be time for your presentation. Great.
Great.
Yeah. Dr. Timothy Whitehead, he's an assistant professor in chemical and biological engineering, and he did his bachelor in chemical engineering with magna cum laude at the Vanderbilt University in Nashville, and his PhD in chemical engineering at the University of California in Berkeley. There his thesis was already about engineering proteins to build nanostructures. His advisor was Douglas Clark, and he won various awards from the College of Engineering nominee, Johansson Crosby Endowed Chair, and Young Scientist Keynote Speaker, and he also won a global award from MSU as a fellow and NSF Career in 2013. So, we really appreciate you taking time to come here. And before, as I said before we start, we kind of think it's interesting to learn about what led you to the path of science, basically. Was it something you were always drawn to? Maybe you had a great teacher that kind of elicited some or sparked curiosity in you. Yeah, what was it that brought you to science? Thank you.
Yeah, so thanks. I mean that's a tough question, right? But I think for me, I wasn't thinking much about science. As an engineering undergraduate student, I graduated and didn't really know what to do with my life. So I was a beach lifeguard, and then I worked in New York as a environmental engineering consultant. And so what we did was we spent a lot of time um, building environmental remediation plans for kind of environmental disasters from kind of the post World War II era. So Lockheed Martin, for example, would dump solvents down a dry well and on Long Island. These kind of things that you would go and clean up. And what I realized pretty soon, designing kind of some of these plants, is that it's um, the technologies that you build to go and the plan is built around don't really solve the problems. They solve the immediate problem of, for example, contaminated groundwater, but they don't solve the long-term problem of how do we get rid of these solvents completely. From once we turn these plants off, the solvents will return into the groundwater. And so I wanted to go to get my PhD and really think about kind of those kind of technological solutions. And that's, of course, very engineering focused. And then in grad school, I kind of really liked the idea of science and in more kind of an open investigation. And so kind of my lab right now, we're in engineering department, but we do very applied technology. And but we do it at the boundary of science, where we learn new things about kind of how proteins work or how systems in biology work.
Yeah, and I think that's always interesting how the path, how the different paths are. And yeah, most of the time it's just curiosity about something, or a specific project, or so. Yeah, I think it's really interesting. And to do this research, I know you already told us a little bit what led you to this type of research, but was there maybe, like, was it easy to get funding for this? Was it maybe something people told you crazy? Basically, we had a lot of speakers telling us that in the beginning, it was a huge struggle, or was that something that was really, came out of the collaboration or an advisor you had? Is there maybe a little bit of a backstory for this specific project? If not, that's also fine. We can skip that. Thank you.
Yeah, so actually there's a little bit of a backstory, but it's part of the presentation. So maybe we'll keep the suspense up and we'll learn about it
Oh,
then. In terms of the actual in terms of the actual funding, we tried we wrote two proposals. One did not get funded, and this one that predominantly funded this work got funded, and since then we've been pretty successful with funding, and that's by no means uh, guaranteed in this kind of line of business. So at one point in my career, I think I had 30 or 35 grants in a row or proposals in a row that were rejected. So it's nice to actually get funding for once for kind of the stuff you like to do,
that's
right?
amazing. That's amazing. Just two. Yeah, I'm more in the 32, right? Like,
Yeah.
usually. No, we had a couple of collaboration projects that got funded pretty easily, but then others are like, okay, that's wonderful. Congratulations. And yeah, please go ahead. The stage is yours. And yeah, we are curious to hear the story. Thank you.
Yeah, so thanks everyone. I'll try to keep this as short as possible to leave as much time for questions as possible, and I'm going to try to go at about 20 minutes or so. And what I like to start off with is just if you can see the slides and kind of go to that first slide here, which is that introduction slide. I'd like to kind of lead off. I think a lot of times in science, it's uh, there's always uh, a masochistic streak where people want to say how much they work and really don't try to go and bring in family. And one of the things for COVID in the last couple of years is it's important to kind of show a personal side. So this is my son who's 4, who the first day the COVID vaccines were open for under fives, he was in line and he got his vaccine, and he just I just got off from lunch, and he got his second shot today, so he's fully boosted in time for his grandparents to come next week. So what I'll tell you really about is kind of a new concept in biosensors, and this is really a Nature Biotech paper that just came out last month, and we use plant hormone receptors to go and create this biosensor platform. Um, so I'm going to go to slide 2, and the slide 2, like what I'd like to start with is acknowledgments, because there's a ton of people that also collaborated on this research, and it's really important not to go and give them shrift at the end. And in particular, the PI for this funded project from DARPA was Meter Nusinoff at the Danforth Center, and then also at University of California, Riverside, both Sean Cutler and Ian Wielden, that you can see the pictures there, or also were instrumental to this research, and in fact, they could give the exact same talk to you guys. And then at my university, PJ Steiner and Matt Bedowitz, who were former postdocs, they now have a startup in stealth mode in San Francisco, and you'll hopefully be hearing about that in a couple of years. They did the predominant amount of research in my group. And so I'm going to predominantly talk about this Nature Biotech paper, but there's also a couple other smaller, less general interest papers that I'll also feed into on this presentation. And then finally, because this is an audience that I don't know many people, this is a great opportunity for us. We are looking for a lab manager, so someone who's just 0 or 2 years out of school and is looking for a gig before they do a PhD or medical school. So for a 1 or 2 year gig. If you're interested in that at all, or you know anyone at all who would be interested in moving to Colorado, Boulder is beautiful. There's amazing mountains right outside my window. Just contact me by email, and my email is pretty easy to find. We also have an opening for a postdoc, but that's less pressing than a lab manager. Okay, so I'm going to go and get to the motivation pitch, and that's kind of the third slide, so slide number 3. And how I like to motivate this is to think about kind of all the ways you could, or in terms of biotechnologies, anything you could do if you had a biosensor or genetically encoded biosensor, so something you could go and put into an organism, and it could go and sense a molecule and respond for nearly any possible small molecule. So no matter the small molecule you can imagine, you could go and build a biosensor for it, and just think about the possibilities for biotechnologies. So you can imagine, prosaically, and this is why DARPA funds this. You can imagine doing this for threat detection. You can imagine toxicology screening, right? You can integrate with to go and find fentanyl or THC in your bloodstream. You can control plant traits in the field. And so what I'm showing you here in this picture is you can control water use with small molecules. You can sense metabolites in real time, and there's a lot of push both in industry and basic science to go and get sensors that can go and sense metabolite fluxes and metabolite concentrations. And then kind of what I'm interested in is more on the applied side, where you can imagine, and in fact, people have published ways to go and engineer microbial species to go and have, for example, dynamic flux of their metabolism in response to small molecules in a pathway. Or you can imagine behavioral modification of living animals, or even engineered cell therapies like CAR T cells. You can imagine spatio-temporal control, so you can turn them on or off at defined portions. So all of these biotechnologies and many more that we probably haven't thought of or invented yet would be enabled if you could go and not be limited to kind of a few chemicals. You could do anything, and so that's kind of where it's really motivating for us and me. And so the challenge, of course, and so I'm going to go to slide 4 here. The challenge is that building these biosensors, this general idea, this pie in the sky idea, it's hard. And it's hard because you have to make them portable to monkeys and microbes. You've got to make them tunable for the concentration range you want to set them. You have to make them general, so general purpose biosensor. And so the next slide gives you different concepts of how you make these sensors. You can imagine making synthetic antibodies or antibody-like domains, or transcription factors, or GPCRs, or these are G protein coupled receptors that are form the basis of much of our sense of smell, for example, or other signaling in the brain, or even aptamers, or using RNA instead of proteins. And the challenge here is that those biosensors require really two things, and any biosensor requires those two things. The first is some kind of molecular recognition event. So you have to recognize the molecule by usually non-covalent binding. And that's generally easy to do. The field of antibody engineering, so a third of my lab does antibody engineering, and there have been decades of advances on how do you engineer antibodies or other antibody-like molecules to bind things. And so the binding event, we've kind of worked out. But the real challenge is that transduction. You have to transduce that binding event to some kind of output. And that's really been the difficult part in generating these biosensors with properties that we would want for nearly any small molecule. And so I'm going to the next slide. And kind of where the story begins for me, the story began for my collaborator, Sean Cutler at UC Riverside about a decade ago. In 2009, Sean's group published the biochemistry and the structural biology of a hormone perception system in plants that senses a small molecule called abscisic acid, or ABA. And this controls a lot of things in plants, including water use. And he's deservedly in the National Academy for that discovery. But he wanted to play around with that receptor about a decade ago. And in 2015, he published a paper in Nature where he showed that you could engineer that receptor to bind to an existing agrochemical called mandipropamide. And when you go and activate that engineered receptor, you can go and imitate the ABA signaling. And what he showed is the transgenic line on the right-hand side, you have two plants here, a wild-type plant, a Arabidopsis, and a plant that has that transgenic MANDI receptor. When you spray the plants with the mandipropamid under simulated drought conditions, the one with the MANDI sensor lives and the wild type dies. Right. And so this was kind of a first demonstration of what I would think of as plant synthetic biology. But what I was really excited about is I thought that this may be a general solution. And of course, to Sean's credit, he probably had that same idea, several years before I did. So what I did is what's nice about being an academic is you can invite people to your institution. And so I invited Sean to my institution at the time, Michigan State, and we had a chat, and we really liked some of the ideas that we had. My background kind of in protein engineering and antibody engineering, and his background in this system. And we thought it'd be really nice to collaborate and develop kind of this platform, use this platform not to sense this one agrochemical, but as a general platform for biosensors. So I'll go to the next slide. And how these biosensors or plant hormones work in general, and so you have to know a little bit about the science about how this perception works, is they control plant traits, and it's fascinating, using something called chemically induced dimerization. And what that means is that the hormone is a small molecule, and I'm showing that in orange here. So whether it's abscisic acid or gibberellin or auxin or osmotic acid, in purple is the receptor protein that binds that small molecule. And then that binding triggers a conformational change in the protein to recruit a second protein in green, which I call a binding protein for this presentation. And that dimerization of those two proteins that is dependent on that hormone is what's responsible for signaling in control for many plant traits. And how this works molecularly is on the right-hand side, there's a cartoon from Hakashima, where you can take the ligand. These hormones typically are nonpolar with some polar bits. There's electrostatic complementarity in the bottom of the receptor protein, and that ligand or hormone fills the pocket, triggering that conformational change or that lid to close around that receptor. So I'll go to the next slide, slide 8, and how this kind of chemically induced dimerization can be exploited in biosensors is that by bringing two proteins together that aren't typically together in a cell, you can do anything like, for example, on the right-hand side, you can control transcriptional activation. So you can fuse one part to a DNA binding domain and the other to an activation domain. And you can turn on gene expression dependent on a small molecule. You can complement split proteins, so you can engineer a protein to basically cleave the protein in half, and those two halves are normally non-functional, but when they are in close proximity, they become functional. And you can complement them by bringing them in close proximity. Or you can also do more complicated things like activate protease signaling pathways by bringing a protease in close proximity to its recognition substrate. So, using just a general system of bringing two proteins together as a function of that small molecule, you can control a lot of biological processes. And so in slide 9, what we were really excited about this system when I started talking to Sean, and Sean, of course, had I don't want to take credit for these ideas because I think Sean really was at the forefront of this. But it's really exciting because this abscisic acid receptor system, this plant hormone receptor, had already been shown to be portable across domains of life. And so Crabtree at Stanford had already shown that this system is functional in mammalian cells. Sean had shown in that Nature paper that you can engineer this receptor and it's functional in plants. And then some work from my lab in yeast as well as in vitro measurements showed that you can basically port this across domains of life. On the next slide, the second really key advantage of this, and this is an engineering type advantage, but it's important. So I'll mention it real quick, is that we did kind of some of the biochemistry, and we can write rate mechanisms for how the system works, where the hormone receptor first binds that small molecule and triggers a conformational change and then recruits a second protein. And what you can do with this system is simply by changing the ratios of one of those two proteins, you can change the effective concentration at which you sense that small molecule. And I'm showing you a model in blue line, and then also experimental data in open circles, and as a function of this extra protein. And so what you can do is you can actually tune really high affinity sensing from relatively low affinity sensors. And that's a key advantage if you really want to sense really small concentrations of molecules or turn on pathways with small molecules. And then finally, this is also an engineering advantage, but it's also key to why I thought this system was going to be so successful. And the key thing is that if you remember, binding is easy, but transduction is hard. And the reason transduction is hard is because there's conformational changes in the protein, and to engineer those is really challenging, much more challenging than molecular recognition. And what we show here is that if you make a ton of mutations at the molecular recognition side, at the binding side, it doesn't interfere with the transduction event. And so that's really important for the generality of these sensors. And so based on these things, we really thought we could develop these sensors. And so kind of on slide 12 is really the question that motivates that Nature Biotech paper. And that's really just one question. And the question we had was that, can these sensors be engineered to bind diverse chemical ligands with affinity and specificity? So if we take all of the decades of experience on antibody engineering to go and really crank down on the binding, can we actually get to sense diverse chemicals? And so on slide 13, I'll show you kind of the meat of what we did in my group. We use something called computational design, so computational protein design, using a macromolecular software package called Rosetta. And what we did is we identified mutations in that binding pocket. So we will make amino acid mutations at that binding pocket to go and change the diversity of the ligands that will be bound in that pocket. And so we did that by computational design, 19 positions that we could. And then the next slide, slide 14, is once we have those sets of mutations, the question is, how do you make that into a set of proteins that we could test? In protein engineering speak, this is called a protein library. And we built that computational design protein library using a couple techniques that we've developed in my research group. Where the meat of it is we call up Agilent or Twist or any synthetic DNA provider. We can order synthetic oligonucleotides containing thousands and tens of thousands of mutations on DNA, and then we can encode that DNA in plasmid encoded libraries using a one-day procedure that Emily Renbeck and my group had invented about 5 years ago. We then transform that library of protein variants into a yeast, baker's yeast, and then we screen it using a technique called yeast-2 hybrid screening, where we how the crux of how that method works is that we can activate a necessary synthetic gene in the presence of our small molecule, and the cells will grow if that synthetic gene is expressed, and they won't grow if it's not expressed. So slide 15 shows kind of the results of those yeast-2 hybrid screening, and also after affinity maturation of some of these sensors. And so I'm first showing you kind of a set of synthetic and natural cannabinoids that we screened and we were able to find sensors for. The blue here is activation of a laxative reporter, and on the top columns are different concentrations of the chemicals. And so we can do everything from CBD oil to THC to some kind of these synthetic cannabinoids you can get in head shops that are marketed under things like K2 or Spice. So I'll go to the next slide. So slide 16. It's not just limited to that class of molecule. We can also do organophosphates. And I'm showing you here a bunch of diverse organophosphates, and for which we can go and identify sensors for. And again, the blue shows the sensitivity of the response for given sensor compounds. These are not affinity matured sensors. You can go and take, for example, I'm showing you structures like pyrimidine or diazinon. You can go and take the initial sensors or receptors for those compounds, and you can use directed evolution to go and get very, very high affinity biosensors. And then the next slide, slide 17, just shows that these sensors are very potent and specific for their intended ligand. And so pyrimephos, methyl, and diazinon differ by only a few key changes. You know, there's an ethyl linkage instead of a methyl linkage in a couple cases. And you can go and I'm showing you results here where there's two sensors, the peer one with the piri and the peer one diazi, and there's no cross specificity. So you can go, if you engineer these sensors, they're specific for their intended chemical ligand. And then slide 18 just shows the portability of these sensors across a number of in vitro and in vivo sensing modulalities. And so, for example, I mentioned before, you can go and do activation of gene expression. On the top right-hand corner, I'm showing you activation of a fluorescent reporter gene in response to small molecules. On the lower left-hand side, I'm showing you reconstitution of split proteins with in this case, luciferase split protein. So in the absence of the ligand, there's low luminescence and there's high luminescence when you add saturated amounts of your ligand. The next slide, which is slide 19, is just showing that these sensors can be used directly in toxicology screens or ELISA-like assays in biologically relevant matrices. So there's a bit of an optimization that we did in my group, and then we went to an expert, Wenwen Zhong at UC Riverside, who does these kind of sensing for a living. And her group showed that these sensors work in saliva, urine, serum, and saline with toxicology relevant limits of detection for these given compounds. Okay, so kind of in the picomolar range. And then Francis Peterson, so in slide 20, Francis solved the structure for one of these sensors and really gave us a good insight into how these sensors are actually working. In blue, I'm showing the biosensor. In orange is the small molecule, the synthetic cannabinoid, which I'll call WIN for shorthand. And then in green is the binding protein HAB1 that comes in. And if you zoom in on the structure, on the details, really the key linchpin is there's a bound water that makes hydrogen bonds with all three molecules, the biosensor, the small molecule itself, and the HAB1 protein. And in fact, we think that water is key to the interaction for these ligands. And in fact, you would expect, if that were the case, to see hydrogen bond acceptors for kind of all of the sensors that we've seen. So, hydrogen bond acceptor in this case would be an oxygen on that naphthoyl. Here, if you remember your organic chemistry, but if you don't, it's just that red molecule where that orange is carbon. And so on slide 22, if you go and you look at the structures of these compounds that we're able to identify, you can see that COO is present in a large majority of the synthetic cannabinoids, and in fact, some of these organophosphates as well. And so we think that key hydrogen bond acceptor on the ligand side is going to be a requirement for kind of really high affinity sensing for many of these compounds. And so I'll end there for that paper. The next slide, 23, I think is kind of philosophically where I'd like to end. And so you can really go in two different directions as a scientist. One is to say, you want a sensor in the community, I'll build you a sensor. Write me in on your grant, write me in on your next R1, and I'll do that. Or what you could do is you could ask, what if hundreds of labs around the world could build their own sensors for their own applications? And so I'm much more in the latter camp of open science, and I really want to be able to democratize the sensor discovery, similar to the democratization of synthetic antibodies a generation ago. And that occurred, Marx's lab at UCSF or Dane Wittrop's lab at MIT or other labs around the world. And so that's where we're at. And I think that I'd like to end there. That way we can leave as much time for questions as possible. But these sensors are on AdGene, so they're freely available to any academic lab worldwide or nonprofits. And then all programs and algorithms that we develop and publish are either on GitHub or they're on the Rosetta Commons for the macromolecular software that we use. And what I'd like to leave kind of as a take home is that really just to mirror what we started with, what biotechnologies are enabled if you could have genetically encoded biosensor for nearly any small molecule. And what I've described today and is described in that Nature Biotech paper is a platform technology from plant hormone receptor for sensing small molecules. And we'd like to push this as far as we can, but we'd also we'd also like the community to go and have a run at these and develop their own sensors. So I'll end here and happy to take questions.
Thank you so much for this wonderful presentation, and especially, also for the ending of your presentation that you want to make this open access, basically a platform for everyone to use. That's remarkable, and thank you for that. And this techno, it can be used for so many different things. So let's start with like a general application for everyone using it at home. Let's say you waste all your money on organic fruits, as a parent especially, and you want to test that. Do you think we will have something based on your platform like that for everyday people, or do you want to like, or is it going to stay like more in a lab research type of environment? Because I think that would be actually really cool. You put a drop on your fruit, and then and then you could see if you wasted your money or if it's actually organic or something like
Yeah.
that.
Yeah, so what you're describing in some cases is like there was a big article in Nature yesterday about things called plasmonic biosensors, where they're using them right now commercially for detecting off tastes in maple syrup in Quebec. And so I think things like that could easily be applied using our sensors with other existing output modalities like these kind of plasmonic sensors. But that's this is why we want to democratize it, because we can't do everything, and we're not that interested in that specific application. But if people can take our sensors and go and apply those for those sensing, then absolutely you could imagine using this kind of platform for things like you're describing.
That's interesting. And so you said this would be also applicable in vivo. So let's say, would you be able, or would one be able to express a protein in the brain and let's say be very good at sensing dopamine levels, other type of neurotransmitter or hormone levels in a living animal model, or maybe in cell culture organoid models?
Yeah, I don't want to give too much away, but one of the big things we're interested in now is porting these to mammalian applications, and we're an engineering group, so we're really key on thinking about how you could apply that, and the obvious the obvious thing is for kind of engineered cells like CAR Ts, where a number of other groups have done small molecule control of CAR T, and they and so you can imagine applying this to that front very very easily. An area that we're interested in that we don't have the expertise in house for is also metabolite sensing, so real time metabolite sensing in living cells and organisms as well. And I think those are going to require kind of collaborators who can do that type of science. But those are kind of the big areas we're thinking of right now.
So a really big one. We had the guest speaker here who did really great discoveries on astrocytes involvement in the brain, how important they are, and that potassium levels are quite important for regulating like upstates in the brain and stuff like that. But it's really hard to measure potassium at those low levels reliably. So if you could please go help work with that to do potassium sensing in the brain, that would be really amazing. But sorry.
Yeah. So yeah. So I mean, I think potassium is tough, right? I think that's so I think this technology is more for like, C6 or high, so 6 carbons or higher. I think for potassium, you really have to think about um, engineering something like how calmodulin has been re-engineered for calcium sensing. You'd have to do that, so it would be a different platform, I think. But yeah, that's not something I've thought too much about, because I think I agree with you and your former speaker how difficult that is to actually go and measure.
Okay, yeah, thank you. And please, Frank, Denise, Dr. Shah, and everyone, go ahead with your questions. And yeah, thanks for answering mine. Thank you.
Yes, thank you so much. That was a wonderful, presentation. And I was just wondering to ask you a little bit of question around the application of that, because you mentioned about the immunosensor. If I'm right, I'm not sure about that. But I was just wondering to ask you, in the case of the cancer therapy, especially conjugated cancer therapy and targeted therapy, did you think about that part or not?
Yeah, we're yeah. So the short answer is yes, and I'm glad to see other people going in the same direction. But we're not. We're very early stages with thinking about exactly what to do, kind of in that space. And what I can tell you is that there's two ways that you can imagine going about it. One is that you could um, well, I'll leave it at that. I think it's early enough stage, and this is this is the stuff that gets published in 2026, right? But yes, we have been thinking about that, and if anyone in the room is interested in those ideas, I'd be happy to follow up, offline, where I can think a little more carefully about what I can say.
Yeah, because of the innate immunity and all of those functional things that we see in the T cells and behavior of the T cells, especially about the tumor and the metastasis of the tumor. And I think that your work can be very helpful in that case. That's why I ask you. Anyway, thank you so much.
Great, thank you.
Frank, Denise, did you have questions so far?
I don't have. Thank you very much for this amazing presentation. Uh, some of the things remind me of other fields, but right now I don't have any particular questions. Thank you. Maybe Denise, you have some.
Thanks, Frank. Hi, Chen. That was a really fascinating presentation. When you mentioned earlier the disposal of solvents in ground wells, that brought me back to undergrad, and so I really appreciated your philosophy and approach on how to, rather than be reactive, be proactive, and being able to get ahead of the problem. I only had one question, and it's probably sort of a very basic one, but jasmonic acid, is that because it was first cataloged in a jasmine plant, or what is up with that name?
Yeah, I gotta be really careful, because my a friend of mine is in the National Academy for discovering this pathway, but I assume it's related to the plant species it was originally discovered from, but I'm not positive on that. Um, I'll you reminded me to send him an email to go and ask him about that later. Thank you.
For sure. I really appreciate the way that you diagram gibberellin and auxin, showing like the protein folds and all that. I don't think people are used to seeing things like that, so I think it's really nice that you included that visualization in the slides.
Yeah, I have to agree. That's one thing. I've seen these things just recently with COVID. So, but the graphics definitely. Just want to underscore that.
So what's if it's okay to ask like general questions that maybe I can think of people maybe in the audience are interested from a general background. Why plant hormone receptors? Like what exactly is the upside in using plant hormone receptors versus other receptors or proteins exactly?
Yeah. The key thing is that 30 years ago, Stuart Schreiber and Crabtree and others um, basically used synthetic proteins um, in a small molecule to go and do chemically induced dimerization, and that has been kind of transformational for the field of chemical biology. And they have 30 years of experience, kind of saying if you take a small molecule and two proteins that don't normally come together, they come together. That chemically induced dimerization. And so you have kind of those decades of transduction mechanisms. And so the key advantage for plant hormone receptors is that they do natural chemically induced dimerization. And I'm sure that exists somewhere else in nature that kind of signaling in response to small molecules. But if there is, it's not as easy to apply as these plants, or it's unknown to me. And so this is kind of the best system where we can take advantage of those decades of chemical biologists going and making new transduction mechanisms for two proteins to come together non-covalently in response to a small molecule. That's, for me, that's.
Sorry, you cut out because I think you got a phone call.
Oh
You
yeah, I
muted.
said for me that's the key advantage that I see.
And then is this so let's say for the consumer later, will this be a visual cue? What's the outreach mechanism that the consumer will use?
Yeah, so we have several right now. For the toxicology screening, which is most mature, it's linked to horseradish peroxidase, so it's an absorbance measurement change. And the reason that we do that is so it's compatible with all of the kind of current toxicology screening that you do, which is kind of plate-based, ELISA type technologies. Um, for uh, for other sensing, I think it's going to depend on the specific application. So one thing I can talk about is um, we're hooking this system up to a fungal bioluminescence pathway. So you can go and control post-translationally, so not having to make new protein, you can control bioluminescence. For the DARPA project, where you put these sensors in plants, they need to be able to go and sense these with drones or remote detection. And so the big things that we're doing are turning on pathways for pigmentation, like anthocyanin, as well as so it'll turn the plant purple, um, as well as controlling things like temperature in the leaf or other measurements, non-fluorescent measurements. So you can actually see by infrared, for example.
Yeah, that's really interesting. I use bioluminescence. Like I used bioluminescence in the brain, but for for actually optogenetics, which is actually also algae based. So that's really interesting that you use these different approaches. For the DARPA project, if you change the color of the leaf, that can that mess with the photosynthesis efficiency, or it doesn't really matter for light absorption?
Uh, what I can disclose, this is not this is not my research, so I'm not sure what has been disclosed and what I can't disclose. Um, what I can disclose is that you could anticipate that it could impact photosystem efficiency. And fortunately, people have published ways to increase uh, um, uh, photosystem efficiency, um, uh, you know, in the last 3 or 4 years. And you can imagine doing something similar to go and counteract those decreases in photosystem efficiency by turning on pigmentation or other things that can absorb photons.
Well, it could also be good, right? If you're supposed to be an organic farmer and you have that stuff in there, it turns them kind of, if you use pesticides, it turns a leaf into something that they are not so efficient. That's really against, against the farmer's interest. Like, you have a double reward by having the plants grow better if you don't use, because, the plants won't turn color. That would be actually
Yeah,
good. Hi, Lisa.
yeah, you can
Sorry, go ahead.
go ahead.
No, I just wanted to say hi to Lisa and if she has a question, but please go ahead first.
Oh no, I think that's yeah. I mean, I think there's a lot of things you can think about with this technology. That's all I was going to say. So definitely, I can take another question.
Yeah, I was actually curious if there were implications possibly for firefighting or intercepting the signals in a way that would be helpful for climate change, or if you have any ideas on that front.
Um, for firefighting, is
Yeah,
that what
for
you're saying?
fire prevention or for like ways of interpreting or intercepting the signal pathway in a natural environment such that we could learn more about the environment we're in and better prepare for droughts or any kind of, climate setbacks that we're facing.
Yeah. So my Um, a big push by my collaborator Sean at UC Riverside is to go and be able to control to make field ready crops, and so the majority of kind of our cash crops now are transgenic, and one of the big things about these receptors is that you could change them minimally with current technologies, and so they would not be classified as transgenic plants. But you could get them to respond to already approved chemicals or agrochemicals. And what he showed in that Nature paper is that you can control crop traits such as water use directly in the field. And why that's really important is because we can see 2 weeks ahead of time with current technologies, weather technologies, but plants typically can't, right? And so if you could go and tell the plant 2 weeks ahead of time to conserve water, then you could keep yields up, and you could control different things in the field in ways you can. And so Sean is really interested in that aspect of for mitigating for climate change. I'm not positive how for fire prevention. I'm not positive how useful these sensors would be, but maybe it's just because I'm not imaginative enough. So I'd be able to like to hear any thoughts on that. I mean, I'm sitting here where I live in Boulder. We had a fire 2 miles away from us that destroyed 1,000 homes, and we had a fire a mile in front of us. So we were sandwiched in the last year on fires on both sides of us that destroyed plenty of homes and property. And so I'd be interested in thinking about that option more.
Yeah, I don't know if you can change, but that would not be with the biosensors. But if you could make the maybe specific plants that you would put around areas that are at risk of drought, maybe change their color if they kind of have less water, like that the leaves change color. But that would be probably a different platform. Another thing what I think would be really interesting is to have plants in cities that should comply with a pollution regulation, air quality regulation for basically the citizens, not just, to have on your iPhone, but to have a real life indicator of pollution levels in city plants, or maybe some bioreactors that would carbon capture, but at the same time change color if there's too much pollution in the air. But something like that, I think, would be beneficial. Maybe not direct climate change, but at least air quality or ozone levels. Would that be possible if ozone levels? No, but that's maybe fluctuating too much throughout the day. So, but yeah, maybe pollution levels. I'm not sure. Is that something, Lisa, that would maybe go in that direction?
Yeah, definitely. And I think too, the awareness of drought and preventing fires is, I mean, a lot of these are started by humans, and maybe if they just have better signals or more clear data, they would be more cautious, one would hope. Yeah, and then there's also been interesting data about crop yields being down due to slight increases in nighttime temperature that was an unexpected potential contributor to food shortages and things having to do with climate change. And so I think there's, probable applications there as well. If you could slightly expand the temperature range or the uh, yield to compensate for an extended range and these kinds of things. But it seems like, yeah, making major breakthroughs. So thanks for coming on and talking about all this.
Yeah, thanks. I appreciate it.
I have a question, Tim, if you have a time
Sure.
still. So my question is about, you know that we have a nanoparticle-based biosensors, and I was just wondering about your sensor have this capacity for analyzing the extracellular vesicular matrix or not, or if you want to compare it with the nanoparticle, how you describe that.
I'm not. Maybe I'm not too familiar with what you're describing. Maybe you. Is it possible to give me a little more detail?
Which part about the nanoparticle-based biosensor?
Yeah. Yeah, the nanoparticle-based sensor.
So normally they are using that. I mean, I'm just talking about my job perspective. They are using for the extracellular vesicle, and sometimes we are using that in a liquid biopsy for, catching some of the vesicles from the tumor. And we have this type of sensors, and they are working very good in extracellular matrix of the tumor, and they can catch the vesicles. And I was just wondering, maybe you have some information related to that that you can compare with your biosensor that you want to develop in the future.
Oh, I see. Yeah, we're not thinking. We're thinking more of um, we're thinking more of either delivering small molecules um, intratumorally, so like just direct injection to the tumor, and then have a diffusion, and then being able to control kind of proliferation of example engineered T cells using that more than vesicles. I think the vesicles, for me, it's I think that that's hard. It's hard to do as an academic. Let's just say that, just because it takes a couple of years of training to really get the procedures down for, at least in my understanding. It's hard to work with those kind of extracellular vesicles. But I could be wrong, but that's just an outsider's perspective. But there's no direct comparison with that technology. No.
And do you think that there is more affinity of your biosensor for the certain organs of the body?
I think we could sense if there were metabolites, so small molecules that were overexpressed relative to, elsewhere, then yes, but it really is specific for kind of those small molecules. And I think the sweet spot are kind of things that look like drugs, and so um, you know like things that could drug another protein or predominantly. Those are the type of things that this system is really well suited to kind of sense.
Yeah, thank you. So could you detect RNAs?
I don't think you. I mean, I think you can detect. I think you would be able to detect individual nucleobases, But you basically have got to envelop the entire molecule in that pocket, and the pocket's not large enough to accommodate, things that are much bigger than kind of one nucleobase.
Oh, okay, I see. Yeah, I was thinking if that would be possible. How many? Well, you could. Well, then you could create a signal. I don't know. Yeah, that would probably not work because if you could, you could have an in-cell microarray if that would be possible. You know what I mean? Like you could inject it during development and then see how color patterns maybe would change during, let's say, migration of neurons to the spinal cord or something like that, and see what's actually important, and then know what to do to get spinal neurons to actually implement in the leisured spine, like in a spine that got hurt by an accident or stuff like that. That would be actually really cool. But yeah, what factors would be important to get neurons, let's say, to implement themselves again in a specifically brain context or neural context? That would be quite a breakthrough. Let's say you use stem cells to save Parkinson patients, like to recreate dopaminergic cells again. You can do that, but they really don't do a good job of integrating themselves into the network. So if you could study that better with centers like yours, that would be quite interesting. But yeah, Suja, welcome to the stage. Do you have a question?
Yes, very, very fascinating conversation. Thank you, Tim and Katerina. I actually had a question specifically regarding the computational design aspect for identifying the combinations scenarios. I actually joined late, so forgive me if this was answered earlier, but did you use ML and AI models to do this? I know that, for example, there's the Folding at Home project that also deals with trying to find the permutations of proteins, right? So I'm just curious more about the computational side of that. Thanks.
Yeah, so we did not. We used a program called Rosetta. So it's a um, all atom, physically realistic macromolecular software program to go and identify um, combinations in a physically realistic force field. The We are applying machine learning to uh, identify what sensors, what molecules we can get sensors for and which we can't, to improve the process. But that is not that work is not ready for prime time. And so, but I can say that the earlier work was not done with machine learning, and we are implementing that into kind of our pipeline. Um,
Very nice. So that software suite that you mentioned, it just did like basic, like heavy lifting computational statistical analysis to find those permutations. Is that right?
so what we did, it you can model different protein designs using that software, and it's physically realistic. So it has van der Waals and directional hydrogen bonding, um, solvation effect, the hydrophobic effect that you learn in Gen Chem and things like that, in a physically realistic manner to go and identify the combinations of those protein sequences. There's a base package, and we had to write our code to get it to do what we want, of course. But it's pretty useful for these kind of design efforts that we use.
Very cool. Thanks. And I'm guessing that code was in R.
It's not actually. It's in C++, and then the there's wrappers. There's a Python wrapper, and then there's an XML wrapper. And for this paper, we used an XML wrapper around the C++ base code.
Okay, very cool. Thank you, and best of luck on your future work.
Great, thanks.
Yeah, does anyone, let me check the, oh, you already answered, already answered the questions in the chat. Thank you for that. I keep having ideas of sensors, but I guess I'm like totally always asking things that are kind of away from your technology platform. I'm really sorry. There's different things I wanted to always do, but they are just not good sensors for it. And I keep trying to ask if you could develop those.
No, that's fine. I think that's kind of why it's interesting to speak to people outside of my field, right? Because it's the best ideas come from pollination and cross fertilization, right?
Yeah.
Across here. So it's great. It's great this little clubhouse that you have set up for talking about science.
Yeah, thank you. So just another thing, a great thing would be like eye motifs and G4. So when DNA kind of makes these little loops of like uh 4 times G and stuff like that, that those are tiny changes that are responsible for like cancer development, but probably also things like neuroplasticity and stuff like that. That would be also really cool if there would be a biosensor for it. Anyways, um, there's one more person that raised their hand. Is that okay? And then we probably have to wrap up because it's an hour now. So, Amy, you have the last question if that's okay, Tim.
Hi, good morning, good afternoon to everyone in the room, and thank you, Tim, for a great presentation. Thank you, Katerina, for having this room today. Um, I kind of came late in the room, so I don't know if this question has been answered. And I'm coming from a layman's perspective because I read your paper and all those. Uh, what you call this abbreviation confused me. So let me ask you this question: how many plant receptors sensors do you have in your list already that you are using at the moment?
Yeah, so I can disclose. So that original paper gave 21 sensors, and we have nearly an order of magnitude more sensors at this stage. And what we're really trying to understand is how far can we push this technology? So this won't be the last salvo or the last paper you ever read on this. But with the acronyms, it is it is what it is,
Yeah,
right? Welcome to biology, right?
I know, right? I was like, okay, I know I studied biology, but I was like, okay, I need to ask him this question from a layman's perspective. But, anyways, uh, I
You know,
have
you know
another
how
question.
we got confused on the acronyms finishing up the paper because like the editors, they copy edit it and they add these acronyms. We got confused on all these acronyms. Like I can't imagine like I'm going to be a cranky old man at this stage, but like it used to be so much easier to read papers. Now you kind of have to open a dictionary side by side with all these papers when you read them, right? I
Yeah, I
certainly
was
have
gonna
to.
Google those, but I said no, I'll just watch the replay and do a little bit more research. I saved your research. But anyway, I love that you have this open source. At the very end of your page, it says that whoever, can approach you guys and talked about it. So are these sensors done in combination at times, or you use one at a time?
Oh, I um, the short answer is that right now they're done one at a time, but I would expect to see something published soon that shows you can do multiple at a time.
Okay, wonderful.
Great.
And when you say small molecule, can you give me like 5 examples of such?
Yeah. Well, melatonin. Let's say tryptophan. Let's say THC. Let's say an organic phosphate that like a nerve gas. And then you could imagine sucrose, right? So those are all examples of small molecules that you can imagine sensing with this kind of platform.
Okay, are these sensors only applicable for plants? Or I know somebody mentioned about cancer. You can use these sensors for the human body?
Yes. You can
Wow.
put them in bacteria, and the bacteria can sense the small molecule. You can put them in plants, and they sense. You can put them in mammalian cells, and they can sense the small molecule. So it's really across the domains of life. We can put them in, in fungi, and they can sense the small molecule.
So,
So if you can transform the organism, you can get these sensors in, and the organisms can go and sense that small molecule and respond in ways that you can engineer.
so let me ask you this question on a layman's term. So, whenever you use these sensors, you don't need any computer to do any result fact finding. It just automatically do what it's supposed to do.
Yeah, well, I mean, if they did what they were supposed to do, all my PhD students would graduate in 2 years. There's some engineering involved, but in terms of the actual the system itself, it's pretty. Um, it's it's the components themselves work as they without any additional engineering. Um, but hooking them up to the transduction and organisms and doing um, the really tight expression that you need and certain organisms is tough, and that takes a little bit of work. But the sensors themselves, they work out of the box, and you can put them in, any. I can't imagine someone who would want to do this, but I'm sure you could put them in an elephant, and it would work as intended.
So, what you're trying to say is, what I'm asking, I guess, can you quantify your results when this occurs? You're
Yes,
able to gather data on it.
yes.
Okay, that's basically wanted to know if it actually works after you implement it. Okay, thank you so much. I appreciate this room.
Thank you, Amy, so much. And thank you so much for asking those questions. They were wonderful. And which reminded me now of a paper we discussed here. I don't know, Dennis, was it like 2 months ago, 3 months ago? It was a paper based on meeting in Switzerland that is held every 2 years about general security issues around the world. And there were pharma companies also present. They should try to, with AI platforms they are currently using, they don't even need supercomputers or anything, with public available data, how fast they could create with those AIs harmful components to humans, more harmful than the most toxic one we have right now available. And uh, I think 3 or 6 hours into doing that, for security, discussion issues, they were able, with public available data, to create 40,000 compounds that were more toxic than the most toxic, like to humans. So, and this was very worrisome, and they made a paper about it. We discussed it. So, your platform, did you ever hear about it? And were you like approached maybe by that? And your platform is probably the closest to be able to address the security issues. I would assume, maybe.
Um,
Like
yeah, I think that I think it's hard. So I'll give like the real world example I'll give is fentanyl, right? And what's on the streets is not kind of, if you Google the molecule fentanyl, it's not what was originally on the streets. It's been changed tremendously, the molecules, and there's a diversity of molecules now to evade regulatory authorities, right? So fluorinated groups have been added, or other groups, to go and change the chemical structure to evade detection, not necessarily to make it more potent, but often to evade detection. Those are very, very difficult. It's very, very difficult to adapt to that Cowden mass game. And I think that's something that my collaborators at Riverside are very interested in doing. But in this biosensor platform, in many ways, is well adapted to what you're describing. But it's something that I'm not particularly that interested in doing. I'd rather spend my life doing other things. Let's say that. So, Katerina, I have a hard stop in a couple minutes, and so maybe we'll stop there.
Yep.
Is that
Yeah,
okay?
sure. Of course. Yeah, that's just reminded me of that, and how fast it would be, your platform to adapt to some new, you know, terror attack maybe or something. But
Yeah.
yeah, thank you so much for coming and for taking the time. This was really, really interesting, and thank you for answering all of our layman's questions, and for your patience. And yeah, thank you everyone for coming and asking great questions. Thank you, Amy, for your questions. They were really great, and I hope you come back one day um, to give us updates when you're allowed to give us updates. And yeah, thank you. This was an amazing room. We really appreciate it.
Yeah, well, thanks everyone for your time, and I really appreciate the questions. And then I just want to reiterate, if you know anyone who's looking for a lab manager position, someone just a couple years, 0 to 2 years out of their undergraduate degree, just let me know, and send me an email. I'd be happy to. We're eagerly trying to find the right candidate for us. And thanks everyone so much for your time. I'm gonna leave now. Okay,
Okay.
bye,
Yeah.
Katerina and
Thank you.
Frank and
Yeah,
all.
we shared it
Bye,
on
team.
Twitter too, through our Twitter account, that you're looking for lab members. So hopefully
Thanks.
something comes. Okay. Bye. Enjoy the weekend. Okay, bye everyone. Thank you for coming. And yeah, we'll have another room later today at 9 p.m. EST. It's a little bit different type of a room. We kind of decided to branch out a little bit about, since we are a high percentage of our team is led by women, and we kind of also want to address issues that women have living in this world. So we have a new we want to maybe try to have a new series talking about listening to stories of women of different sensitive topics, abuse, abortion, and so on, and just having women here as guest speakers, just like our other science guest speakers, and listening to their stories, and maybe help drive awareness and change like we try to do it with science. And we'll have our first room tonight with Ebony. She wrote a book about her story, how of her story of being abused, and how she managed to get out of that situation. And she also founded organization that she now helps women with getting out of these situations, pay for lawyers, and so on. So yeah, if you're interested in just being here and learning from real stories from people, come to our room tonight at 9 p.m. EST. And the titles, when you see titles like this, Doctor someone, and then the title that's very similar to their paper, it will still be always a real researcher sharing their research. And then we, from now on, we'll have, as we had before when we are talking about the book author, in the title will be book author discussion, and then maybe something, a title about their book, or this time it's sensitive stories, talking with Ebony Smith. Just that you're aware that we are branching out a little bit into, we try, topics that are close to our heart and we want to support. So yeah, thank you for coming and thanks for being part of this. And we wish you all a wonderful Friday and happy weekend. Thank you, everyone.